shall learn the female reproductive part gynoecium of a flower. This gynoecium plays a very important role in the pre-fertilization events or changes involved in the sexual reproduction of a flower. So gynoecium, as we have learned earlier, it is made up of, or it consists of a part of pistil. A pistil has, as we know, ovary, style and then stigma. And ovary has many ovules inside it. As we are seeing, as I am showing here, it has many ovules inside it. So now we shall see how the structure of ovule is there. In a diagrammatic structure of ovule, we are going to see here now. Our ovule has this ovule, it has a funicle. Stalk of the ovule is called as a funicle, and the part of the funicle which is attached with the body of the ovule is called as a raphe. And in an inverted ovule there is a ridge or a scar that we call it as hilum. So this ovule, this ovule has a chalazal end. Chalazal end is a part where the funiculus, the integuments and the nucellus meet. So we shall see one by one what all these means. Integument. Integument is a layer of layers which covers the nucellus. Nucellus is a nutritive tissue found, parenchymatous cells, nutritive tissue found inside, and this nucellus is totally covered by layers, and this is called as the integument. If the ovule has single integument, it will be called as unitegmic, and if it has two integuments, it will be called as bitegmic ovule. Then inside it has an embryo sac. See, as I'm showing here, this is an embryo sac. Embryo sac is a female gametophyte. It is a female gametophyte. This embryo sac develops from a megasperm mother cell in the nucellus, that we shall learn later on. The embryo sac, this embryo sac, and here only the egg cell will be there, and the tip of this ovule is not covered by the integuments, and therefore this has a pore, and this is called as a micropyle opening through which the male gamete will enter in. So this pore is called as a micropyle, and opposite to the micropyle is the chalazal end. So this is the picture that we are showing, that is a microscopic structure of an ovule that we are seeing here. Ovules are classified based on orientation, form and position of micropyle with respect to funicle and chalaza. If the micropyle, chalaza and the funicle lie on the same line, it will be called as orthotropous, seen in many Polygonaceae family plants. Anatropous if the micropyle lies close to the funicle. Found in dicots and monocots, this type of ovule. Hemianatropous, if the micropyle lies at right angles to the funicle, it will be hemianatropous. This type of ovule is seen in Primulaceae family members. Campylotropous, if the body of the ovule is bean shaped, it is called as campylotropous, seen in Leguminaceae plant family members. Amphitropous, if the chalaza lies, or the chalaza here it is, see, it is horseshoe shaped, that is the ovule is horseshoe shaped, it will be called as amphitropous, seen in Alismataceae family members. Thersinotropous, seen in Cactaceae family plants, the funicle is too long, and therefore the body of the ovule, see, it is totally inverted inside, and the funicle, if it's long, this type of ovule is called as Thersinotropous. These are some kinds of ovules that we see in different family members. The development of the ovule starts from a nucellus cell which becomes enlarged and functions as an archisporial cell. This archisporial cell, see here, these are the integuments and the micropyle region. The archisporial cell divides into a primary parietal cell outside and a sporogenous cell inside. Primary parietal cell mitotically divides and pushes the sporogenous cell deep inside the nucellus. The sporogenous cell divides meiotically to form 4 haploid megaspores, and this process of sporogenous cell to haploid megaspores is called as megasporogenesis. And these 4 megaspores, most of the angiosperms, only one functions into and completely matures into embryo sac called as monosporic embryo sac. If 2 megaspores they mature into an embryo sac, it will be called as bisporic embryo sac, and if it is for all the 4 megaspores, they develop into mature embryo sac, that type of development will be called as tetrasporic embryo sac development. One functional, see here we are seeing that only one functional megaspore, it is dividing into two nucleus, 2 into 4, 4 into 8. And of these four nucleus, one will move into the center and function as polar nuclei, and these three near the micropyle region, they function as egg apparatus. And of these four near the chalaza end, of this one will move into the center and function as polar nuclei, and these three will function as the antipodal cells. This is the development of embryo sac. Here we are seeing a mature embryo sac, 7 celled and 8 nucleate stage. Here we are seeing 1, 2, 3, 4, 5 and 6, 7, 8 nucleate, but 7, 3 plus 3, and this together it is called a central cell. So now in a mature embryo sac, a pollen tube, through the pollen tube, male gamete will enter and fuse with the egg cell. So irrespective of how the pollen grains will fall on the stigma, and a pollen tube will grow. Irrespective of the place of entry of the pollen tube, it has to go through the micropyle only. If the pollen tube directly enters through the micropyle, it will be called as sporogamy. If the pollen tube enters through the chalaza, but it has to be guided to the micropyle to fuse with the egg cell, this is brought about by an obturator. And if the pollen tube enters through the integuments, it will be called as mesogamy, and then it is guided to the micropyle. So these are the three ways through which the pollen tube enters into a ovule. Next, after the male gamete enters into the ovule through the micropyle, fertilization has to take place. So in flowering plants, the fertilization is called as double fertilization because two ways fertilize the male gametes will fuse. So as we have learned earlier that the pollen tube, there are two male gametes. One male gamete will enter through the micropyle and fuse with the egg cell, and it is going to form a zygote. That will be diploid because egg cell is also haploid, sperm cell is haploid, and the first diploid cell will be the zygote. The other male gamete will fuse with the central cell. See, the central cell is having diploid two nucleus, and this male gamete, so therefore it will form a triploid nucleate cell, and that will function as an endosperm. Endosperm is the tissue which is going to feed the growing embryo. So embryo is diploid and endosperm is triploid. So these two fertilization, that is the two male gametes, fuse one with the egg cell, the other with the central nuclei, and therefore this type of fertilization is called as double fertilization, leading into triploid fusion of the endosperm and a syngamy, the fertilization of the egg cell and the sperm cell, leading to syngamy. So this double fertilization is a very characteristic feature seen in angiosperms.